Hey everyone, we're going to get started with the last session in this main room. I'm Brian Reaney. I'm from Vanderbilt. I've had the pleasure for the last now 5 years of being the dean of what's called the Academy of Kidney Cancer Investigators, which is a DoD-funded virtual academy. And each year, KCA has been nice enough to let the newest class of mentees present their work, present their projects. And there's grants given to mentor-mentee pairs every year that enter the academy for a 4-year term. And again, KCA has been gracious to let them present their data each year. I'm just going to give a couple slides as sort of a very brief background of the academy and some of the accomplishments. And then I'm going to turn it over to them for their individual presentations. So anyway, the Academy of Kidney Cancer Investigators, again, the DoD, which has gracious funding for kidney cancer, about 5 years ago decided to fund this virtual academy. There's actually one in ovarian cancer that's been ongoing for 10 years or so, and they decided to replicate it in kidney cancer, and that's sort of how this whole thing started. And we basically set forth to increase the kidney cancer community, right? And we've talked a lot about mentoring in various sessions at this meeting, and this is really meant to extend mentoring. Most of the mentees are basic and translational. And so it's sort of different mentoring circles. There's a KCA program now, there's this program, there's various programs, obviously institutional mentoring. So it's really meant to complement existing institutional mentoring and bring people together. We have quarterly meetings where the mentees present their projects and get feedback. I have an advisory panel. Celeste Simon, who's sitting here, is on my advisory panel, Jim Brugarelis, Billy Kim, Samara, Turaljic, and others, and people have been in the past, and I'm really grateful for their participation. Active participation in the mentees. I just resubmitted this grant, so if any of you are on the panel in the room, thank you for your kind scoring. And it was nice to put together because it gave me a chance to put together all the accomplishments, the papers, the grants, a lot of the collaborations. Probably one of the most satisfying things that's come out of this academy is the intra-academy collaboration among mentees, between the advisory board and the mentees, et cetera. So it's really been, a gratifying way to sort of enrich the kidney cancer community. I'm assuming I still don't have slides. Oh, they're coming. Well, now I've pretty much said everything, so no. And there are now 5 classes. We just graduated our first class, which was David Braun, who you heard as a rising star, Abhishek Chakraborty, and Lucas Salas. I see Lucas. I know some of the others are in the room. So there was no cap and gown ceremony, but it was great to sort of now see them. And now I know David at least is serving as mentor to one of the people. So I think there's going to be sort of a family nature to this as people graduate and then return as mentees and as frankly as advisory board members. I think at this point I can just sort of turn it over because I've already gone over all my slides. So our first speaker is Aguirre de Cubas. He's going to talk to us about SETD2 deficiency. And then we'll take a question or two at the end. There's four mentees' presentations are about 10 minutes, and then we'll just do a question or two at the end. If anybody has questions, just come up to the mic. Thanks.
Well, thank you for that introduction. I'd like to thank the KCA for having me share a little bit of my work here, and also the KCRP for selecting my grant for funding. So a little bit of background on SETD2. So SETD2 is a histone methyltransferase. It's mutated in approximately 25% of all RCCs and 5% of all solid malignancies. And it's also associated with worse prognosis and aggressive disease. Thus, there's an urgent clinical need to develop therapeutic strategies specific to SETD2 mutant tumors. So to do this, we started off by performing a high-throughput small molecule screen, which identified a BCL-XL inhibitor called A133-1852 as a potential target for SETD2 mutant tumors. We initially identified it in the 7860 cell line and subsequently validated it in several other RCC cell lines where we knocked out SETD2 or added it back in the case of the A498 cell line. We also validated that A133-1852 treatment significantly reduced tumor weights in an in vivo study and significantly extended progression-free or survival in the mice. So a little bit of background on BCL-XL. So BCL-XL is a part of the BCL2 family of anti-apoptotic proteins, which function to inhibit apoptotic effectors back and backs. On top of this, there's a series of BH3-only apoptotic activators and sensitizers, which can either promote apoptosis by directly influencing back and backs oligomerization or inhibiting BCL2 anti-apoptotic family members. So since cancer cells are under a considerable amount of metabolic stress or genotoxic stress, they're usually teetering on the verge of apoptosis and largely depend on this pathway for survival. However, when the balance gets tipped towards apoptosis, back and backs oligomerize, forming pores on the outer mitochondrial membrane, leading to cytochrome C release and unleashing the apoptotic cascade. So, as we all do when we're trying to figure out what's going on in a system, we performed RNA-seq, which showed that there was activation of inflammatory signaling pathways rather than showing apoptotic pathways in our data, which we thought was interesting. So we looked at STAT1 phosphorylation in several RCC cell lines treated with and without A133 and observed that at baseline, SETD2 deficient tumors had primed inflammatory signaling, and when you treated these cells with A133, it agonized inflammatory signaling. We also observed that there was an upregulation of NOXA in these tumors. However, the implication of NOXA wasn't readily apparent, since NOXA doesn't actually interact with BCL-XL, but rather functions to inhibit the anti-apoptotic function of another BCL2 family protein called MCL1. So to determine whether NOXA was involved in the sensitivity to A133 that we were observing, we knocked it out using CRISPR-Cas9 and observed that NOXA knockout completely rescued A133/1852 induced cell death in SETD2 deficient cells. This led us to hypothesize that when you inhibit BCL-XL, the tumor cells are switching their dependence to MCL1. So to test this, we performed dynamic BH3 profiling, which allows us to assess how a stress or a treatment affects apoptotic dependencies in a given cell. So for this, we pre-treated the cells with A133, and then after that, treated them with a series of peptides mimicking BH3 active motifs. And I'm just showing the important part of that data here. So in untreated cells, when we treat with NOXA peptide, there's little to no cytochrome C release, which is an indicator of cell death. However, when we treat these cells, or pre-treat these cells with the BCL-XL inhibitor, and then after treat with the NOXA peptides, we see that there's a robust release of cytochrome C in these cells. Now, you might be wondering why in the wild-type cells we're also observing cytochrome C release, and that's because the only real difference between the wild-type and the knockouts is the knockouts upregulate NOXA while the wild-types don't. And in this system, we're artificially introducing the NOXA peptide in both cell lines, so the functional effect is the same. So this data shows that these cells are switching their dependency to MCL1 when BCL-XL is no longer available. Then we wanted to know what was activating NOXA expression in the system, and it was previously described that NOXA is induced by P53, but it can also be induced by other P53 independent mechanisms such as IRF3 and 7. So we knocked out a series of targets, but in that process we found that when we knocked out CGAS, STING, and TBK1, we saw that there was a rescue of not only phosphostat expression, but also of NOXA upregulation in these cells. These data show that CGAS STING signaling is necessary for A133-induced cell death, but also suggest that the presence of cytoplasmic DNA is necessary for activation of this pathway. So in conclusion, I showed that SETD2 deficient RCC cells are sensitive to BCL-XL inhibition. We also saw that SETD2 lost primes inflammatory signaling in RCC cells. We also showed that cells become dependent on MCL1 when BCL-XL is inhibited, and BCL-XL inhibition activates CGAS STING pathway to induce NOXA expression. And finally, NOXA neutralizes the anti-apoptotic function of MCL1 to cause cell death. However, there's several questions that remain here. For example, what is activating the CGAS STING pathway, or whether or not SETD2 has an immunostimulatory or an immunosuppressive effect in vivo. So based on those observations and these questions, we hypothesized that SETD2 deficiency, or the focus of my grant is based on this hypothesis that SETD2 deficiency primes interferon signaling through the STING TBK1 pathway to confer sensitivity to BCL-XL inhibition, and that targeting BCL-XL provides an opportunity to improve response to immune checkpoint blockade therapy and SETD2 deficient RCCs. So the first aim of this project, we're going to establish the molecular mechanism governing primed interferon signaling and sensitivity to BCL-XL inhibition in SETD2 deficient cells. So in the first part of this aim, we're going to systematically determine the source of cytoplasmic DNAs activating the CGAS STING pathway. And then in the second part of this aim, we're going to determine which one of the IRF 3 or 7 factors is necessary for induction of NOx in the system. And then in the second aim of my project is to establish the role of SETD2 deficiency and BCL-XL inhibition in the tumor microenvironment. So in the first part of this aim, we're going to perform single-cell sequencing to determine how SETD2 deficiency reshapes the tumor microenvironment. Then we're going to perform functional studies in immune cells to determine how SETD2 deficiency affects their functionality. And then in the second part of this aim, we're going to perform preclinical studies using the LVRCC67 cell line that has SETD2 or does not have SETD2. And the tumors will be implanted into mice, and then we'll treat mice with A133 or vehicle, and then after that, randomize the mice into anti-PD-1 or IgG treatment groups. And then after that, we're going to assay a host of variables here to determine if the combination therapy is effective. And I'd like to thank the Academy of Kidney Cancer Investigators, also the KCRP, and my previous mentors, and my current lab and previous lab members.
Thank you. Time for a question or two. If anybody has a question, just go up to the mic. Celeste?
I thought we were going to do them at the end.
No, we'll do them after each, yeah.
Okay.
I didn't think of this yesterday, but what's your guess about the source of the DNA? Do you think you're eliciting mitochondrial dysfunction to the extent that, and there's things, if TFAM expression's changing, you could be release of mitochondrial DNA. Do you think you're causing, obviously there's going to be global changes in, the status of the epigenome, and is that promoting DNA damage and lagging chromosomes? I mean, what do you think?
So initially we thought it was SETD2 loss was causing DNA damage, which was then being agonized by BCL-XL inhibition in some way to cause micronuclei formation. But now we're thinking it's more an effect at the mitochondria. But it's still kind of unusual because apoptosis is considered an immunogenically silent form of cell death. So it's not clear why in the SETD2 deficient background we observe activation of inflammatory signaling. But yeah, we're working on that now.
Do you see changes in, for example, TFAM expression? Because that is meant to avoid mitochondrial DNA release, as I understand it. And actually, Jerry Shadle has shown that, in the mice that are heterozygous for TFAM, they have almost tonic CGAS STING activation.
Well, at least at the RNA-seq level, there's not a difference in expression of TFAM, but maybe at the protein level. Right now, we're trying to perform some microscopy studies where we have a fluorescently labeled TFAM to see whether it goes whether it goes outside the mitochondria when we treat with the drug.
Great, thanks.
Thank you.
Thanks, Les. All right, I think we'll move on to our next speaker. David Schoenfeld is going to talk about a decoy-resistant IL-18 in RCC. David.
See if I can figure out how to use this. Okay. Well, thank you for the opportunity to speak. So today I'll be talking to you about the therapeutic potential of the IL-18 pathway in RCC, including some preclinical evidence of the decoy-resistant IL-18 compound plus immune checkpoints in RCC, and then go over my aims. So just a little bit about IL-18 biology. So IL-18 is a key pro-inflammatory cytokine which activates antigen-experienced CD8 cells and NK cells and can inhibit Tregs. So that's shown here. IL-18 is binding to the IL-18 receptor. It's a potent inducer of interferon gamma. In fact, it was a first-term interferon gamma inducing factor. But administration of IL-18 as a cancer therapeutic was unsuccessful, and this was largely due, in the end, to the secretion of a high-affinity decoy receptor or blocking protein called IL-18BP that blocks IL-18 activity. So interferon gamma, in the end, induces IL-18BP, which then can block and bind IL-18, so prevent it from binding to the IL-18 receptor, inhibiting it, and in effect function as a secreted checkpoint protein. We can think of it as an alternative, an analogous pathway to the PD-1, PD-L1 axis. And this was really demonstrated by Aaron Ring's group a number of years ago when he was at Yale, really showing that it was functioning in this method. And then he went on to develop a decoy-resistant form of IL-18 that was called, that they named DR18, and they used directed evolution to do this. And this DR18 compound could still bind to the IL-18 receptor with high affinity and serve as an agonist and immune stimulant, but was now resistant to binding to the blocking protein, to the IL-18BP. And through this evasion, it could still serve as a potent immune activator and a potential anti-cancer therapeutic. They then went on to show its evidence in preclinical models, including here, for example, a melanoma model called Yummer 1.7, where they showed that the monotherapy of this IL-18 decoy-resistant compound was roughly as effective as checkpoints, and that there was potential synergy when it was combined with anti-PD-1. They then did a lot of really elegant and extensive mechanistic work to show how it was functioning in the tumor immune microenvironment. I won't go into it in detail, but just to summarize, they saw that this IL-18 decoy receptor compound was acting through the receptor of IL-18, it was acting through interferon gamma and also CD8-positive T cells, led to the expansion of intratumoral stem-like and polyfunctional CD8-positive T cells. It can even work, though, in the MHC1 class 1 deficient setting, a very prominent mechanism resistant to checkpoints through the stimulation of NK cells, and it also led to the formation of pro-inflammatory anti-tumor macrophages. So we thought this compound could be very interesting to test in RCC, not only because of the historical significance of cytokine therapies in RCC, such as high-dose IL-2, but also when we looked at the TCGA pan-cancer atlas, we saw that clear cell RCC had among the highest expression of the cancer types included of the IL-18 receptor and the IL-18 blocking protein, IL-18BP. We also saw that in clear cell RCC, IL-18BP was associated with many poor prognostic features, was associated with worse overall survival, but it was also, when we looked at gene expression differences for high IL-18BP tumors, it was associated with gene sets related to cytokine and interferon gamma signaling, T cell activation, and immune response, which was a little counterintuitive. But we also saw that was highly correlated. Among the top correlated genes were numerous immune checkpoints, including the ones listed here, to me suggesting that these are inflamed tumors that then induce a negative feedback mechanism and tamp down the immune system. So we also wanted to see if there was a connection to responsiveness or some predictive qualities to responsiveness to immune checkpoints. So we used a tissue microarray of RCC patients treated with immune checkpoints, and when we stratified based on IL-18BP expression in the tumor microenvironment, we indeed saw that high IL-18BP expressors had worse overall survival. And then when we looked at IL-18BP expression pre and post-treatment by responder and non-responders, we saw no difference pre-treatment, but then post-treatment, the non-responding patients had a significant increase in their levels of IL-18BP. We saw that this was also the case in circulating IL-18BP levels. So this is in a separate cohort of Ipi-Nivo first-line treated RCC patients. And again, we found similar findings, that the responders had no significant increase in their IL-18BP levels pre and post-treatment, whereas the non-responders had a significant increase in the IL-18BP levels post-treatment. This is just another way of looking at that with the ratio of post and pre, again, just illustrating this effect. So overall, these data suggest that the IL-18 pathway may be poised for reactivation in clear cell RCC with a compound like this DR18 that can bypass IL-18BP inhibition. So then we wanted to test this idea in mouse models. So we used the two existing immunocompetent syngeneic mouse RCC models that we had available to us at the time, the RANKA model and the RAG model, not a RAG immune deficient mice, just a RAG RCC model. We wanted to test this DR18 compound in combination with mono and dual checkpoints, so both PD-1 and CTLA-4, and this is our schema for treatment. And then here are some of our results. So we look in the RANKA model. Here's the Kaplan-Meier survival curve. We see the control treatment in yellow, and we see modest anti-tumor effects with the DR18 monotherapy shown in red, roughly equivalent to the checkpoint monotherapy agents. I think interestingly, though, in the blue lines, we saw that there was no additional benefit when we added anti-PD-1 to this DR18 compound. However, and somewhat surprisingly, we saw that when we added CTLA-4 to DR18, we saw a significant anti-tumor benefit, and that's shown in the dark green line on the top. And it was roughly equivalent to doing the triple combination. When we looked in the RAG model, we saw roughly similar results, again, modest anti-tumor effects from monotherapy with this IL-18 decoy receptor compound, but significant anti-tumor effects when it was combined with CTLA-4, but not PD-1, which was not the same result that was found in the other models, melanoma models, colorectal models. So at least in these RCC models, it seemed to be PD-1 seemed to be not beneficial when combined with DR18. So we then did some studies to determine the cellular mechanism or cellular basis of this anti-tumor immunity of the combination of DR18 with anti-CTLA-4. And just to summarize some of our findings, we used immune depletion and neutralization studies to find that the combination was effective and relied upon CD8-positive T cells, NK cells, and interferon gamma, but was not really reliant on CD4-positive T cells. We also did cytokine and chemokine profiling and saw rapid and significant increases in interferon gamma, CXCL10 and CXCL9, and then later on broad increases in inflammatory cytokines. And then lastly, we did single-cell RNA and TCR sequencing, the tumor microenvironment, and saw enrichment and clonal expansion of effector CD8-positive T cells, limitation of Treg expansion, and a shift toward pro-inflammatory anti-tumor myeloid populations, including some interesting interferon-stimulated neutrophil subtypes that we think are important. So I think some key unanswered questions, though, from these initial studies are how did the findings in these models that we use, the RANK and RAG models, translate into more human-relevant mouse models of RCC, including the LVRCC67 model that we had heard mentioned in the previous talk. Also, why is this combination more effective with anti-CTLA-4 compared to anti-PD-1 in the models that we used? And we're giving the checkpoints with this decoy-resistant IL-18 in different sequences, such as priming with the DR18 efficacy. And then lastly, and probably most importantly, how translatable are these findings into humans, or at least human systems? So that leads me to my project aims. So I'll go through them each briefly. So aim one is to determine the impact of combining different ICIs with DR18 on the tumor microenvironment in different murine models of RCC, starting with this LVRCC67 model that was recently developed by Ari Kimi's group. Just to summarize for those who don't know, it involves the loss of VHL, P53, and RB, and the overexpression of MYC. So it's a better genetic representation of RCC compared to the previous models, but still not perfect. It's thought to resemble a myeloid subtype of clear cell RCC. It's very aggressive, spontaneously metastasizes to the lung and liver, and it's generally resistant to anti-PD-1. So we first want to test these combination regimens in this model and then further explore and really try to tease out changes to the tumor microenvironment using different modalities and to help determine why DR18 might pair better with some checkpoints rather than others. For aim two, we also want to assess the efficacy of sequencing treatment with DR18 and immune checkpoints in murine models. And this was based on some of the single-cell findings and some of the efficacy findings. We saw that the triple regimen may actually be worse than the doublet and may be leading to early exhaustion. So we would like to try two different strategies where we prime with the DR18, so give it first and then give checkpoints, or more of a maintenance strategy where we give DR18 with anti-CTLA-4 for a few cycles and then transition to giving alternative immune checkpoints. And lastly, we want to define the effects of combining different ICIs with DR18 on tumor cell death in the tumor microenvironment using ex vivo patient-derived tumor model systems. And I intend to use a model system that David Braun has been developing and working on, where you take nephrectomy or metastectomy RCC samples, fragment them, and put them into a matrix, and then can treat in the solution with different compounds. And then we'll explore the effects on the TME. So my take-home messages are cytokine-based therapies have historical relevance in RCC and are an emerging area of preclinical and clinical study with numerous ongoing phase 1 and 2 studies, and that the decoy-resistant IL-18, or just the IL-18 pathway targeting in general, because there are other alternative strategies to hit this pathway, has therapeutic potential in RCC and other cancers as well, and work is ongoing to investigate the best combination strategies. And at that, I'd like to thank my mentor. My primary mentor is Harry Kluger, and my other mentor is David Braun, and all the people who have helped contribute to this work. And at that, I'll take any questions.
All right, thanks. There's questions. Just jump up to the mic. I have a question I meant to ask you yesterday. For other cytokines, are there binding proteins? I mean, is that phenomenon across all cytokines? Is there something you could learn from other other cytokines?
That's a really interesting question. Something, I was very interested in when I first got involved with exploring this compound. So there are other ways of turning off compounds and other feedback mechanisms, but to my knowledge, this is the only secreted binding protein that blocks a cytokine like this. So it is a little unique in that respect, at least to my knowledge.
Yeah. Okay, any other questions?
Brian, here.
Nazar.
Yeah, thank you. Great presentation, David. From the biology of IL-18, do you have more? Does it have more than one subunit that the decoy can trap one subunit more than the other, or it's just?
So I think the receptor has two subunits that come together and make a heterodimer. IL-18, I think, is just a single subunit.
Okay. The other thing about, was there any synergy of the IL-18 with specifically more with anti-CTLA-4 than with an anti-PD-1, or are you just trying now the anti-CTLA-4 and then subsequently you're going to, in your aims, do anti-PD-1 or make combination of anti-PD-1 anti-CTLA-4?
So
I mean, is there something specific about CTLA-4 and IL-18 that you think you can harness that synergy more than the other immune checkpoint?
Well, that's what I intend to explore a little more. I mean, we definitely saw, at least in the rank and rag models, pretty consistently that it combined better with CTLA-4 and gave more anti-tumor efficacy. We have some initial clues why this could be going on. One is Treg depletion, or at least Treg limitation. We do see DR18 leading to the expansion of Tregs, and we don't see this when you add CTLA-4. I expect that this won't happen with PD-1 either, but we're in the process of confirming that. So that could be one mechanism.
That's great, thank you. All right, next speaker, Rainier Hernandez from Wisconsin. He's going to talk to us about CA9-targeted radiopharmaceuticals.
All right, thank you for the introduction. Rainier Hernandez, as Brian mentioned, from University of Wisconsin. I would like to start by saying thanks to the KCA for the opportunity to present and the DOD for supporting this project. So we're going to be talking here a little bit about drug development on the CA9 space, particularly in the development of radiopharmaceuticals for kidney cancer theranostics. A little bit of a background on what theranostics, we mean by theranostics, and we essentially what we are trying to harness here are receptors that are specifically expressing cancer cells, and then develop agents that can target those receptors. I use those agents as vehicles to deliver radiation selectively to the tumors. And in those vehicles, we can use different types of radiation, some radionuclides that are more for nuclear medicine, for example, PET-CT and SPECT-CT, and where we can target the tumors and visualize those using nuclear medicine techniques like we're seeing here in this kind of example. But we can load up these agents also with radionuclides that decay by a different mode by emitting beta or alpha particles, or with the primary intention of delivering cytotoxic radiation. And this is an example, for example, that this patient got treated with an actinium PSMA. This is a metastatic castrate-resistant